morning, everybody. This time we are going to start talking about the defense against the exogenous causes of diseases. In our earlier lecture, we had talked about the causes of internal diseases and the methods to prevent the internal diseases. The diseases which are being caused by internal reasons. So we submitted that defense not only pertains to the diseases which are being caused by the exogenous factors, but at the same time can also imply for the diseases which are being caused by the internal factors. And we discussed about this beautifully in our earlier lecture. This time we are going to talk about the conventional concept of defense, that is immunity, which is most oftenly talked in reference of exogenous causes of diseases. So, what are the roots of the exogenous diseases? From where the exogenous diseases usually come? We know that there are 4 common external causes of diseases. Ayurveda has beautifully explained these causes under the head of the 𝐎𝐻𝐿𝐺 𝐕𝐢𝘬𝘐, and it defines, Charak beautifully defines that 𝐎𝐻𝐿𝐺 could be rooted through bhuta, visha, vayu, agni or samprahar. So we can summarize them as the traumatic causes of the diseases, that is from agni samprahar adi samutha, or the diseases which are rising because of exposure to the fire or exposure to the trauma. It could also be because of the pollution related causes of the diseases from vayu, or beautifully Charak identifies that there can be microbial causes of disease as well, that is bhutam. And the final cause of external disease or the exogenous root of disease could be the poisoning or vish. Now, this is a very, very surprising revelation these days that we are basically a complex combination of huge sum of living material. We are never alone. We are not living alone. We live in conjunction with so many other living organisms around. Many of these organisms we are able to see, which are macroorganisms or macrocellular organisms, and there are huge number of organisms which are microcellular, and we are living in connection to them. So we are not living alone. So if you look at yourself, you would be finding that there is an environment outside you. And at the same time, you would also be surprised to see that there is an environment inside you. There is a micro environment which is inside you, and there is a macro environment which is outside you. And these both environments are full of microbiota. The microbiota which is there in association with us is called human microbiota. It lives on your skin, it lives on your body, on your surface, and it lives inside your body as well. It is so complex, this human microbiota is so complex that it comprises a whole big structure of genetic material which is now being considered as human microbiome. A big project is being run in USA, which is being called as Human Microbiome Project. And this identifies that there are around 100 trillion bacteria live in and on our body. These bacteria are about 1.3 times more than total cells we have in our body. 95% of these live in our GI tract. GI microbiota can weigh up to 2 kg in a normal person. That means if you are weighing 60 kgs, your original weight is 58 only, because 2 kg is the microbiota which you are carrying. And it has 150 times more genetic material than our genome. Moreover, over 90% diseases can be linked with gut microbiome dysgenesis. This is a very, very important revelation these days, that over 90% of the diseases are being identified to be linked with gut microbiome dysgenesis somehow. And moreover, one very important another revelation is that every individual has a unique gut microbiota, which is as unique as our own fingerprint. This we'll be talking further, because this very point may have lot of implications in relation to Ayurveda, the Agni and Ama formation, and even for the Prakriti identification. So, further talking about the human microbiome, we see that there are more than 1,000 species living in and out of our body, and this forms over 5 lakh type of metabolites in our body. And you will be surprised to know that around 30% metabolites which are found in our blood are primarily derived from the gut microbiome. So you can imagine that how important these gut microbiome are. So finally, if we have to say who we are a mosaic of microbes, both inside and out. Well, now you know that what the microbes are. Microbes are the living beings. They are as living as we are. So when they are living, and they are living within us or outside us, what they might be doing? They might be competing with us for all resources which are around and inside us. They will be competing with us for space, for nutrients, for growth. They are heterotrophs. You know, microbes are heterotrophs. They cannot produce their own food, so they have to rely upon ourselves or on the host for getting the nutrition or for getting the food. If they serve us for getting these benefits from us, they are being called as symbiotic bacteria. So a lot of gut bacteria which are lying inside our gut or which reside inside our gut, they take the nutrition from our body, from our own nutrients, but in lieu of that, they also share some good benefits to our body. So for that reason, we allow them to live with us. So these bacteria, which are relatively helpful to us, are being called as symbiotic bacteria. But besides that, there are too many other bacteria which simply exploit us without giving us any benefit in return. We call them pathogens. These are the pathogens, and we fight with them and drag them out from our body. This is where the defense from the pathogens starts acting. So how do we defend our body from these pathogens? The primary defense could be at the entry point. So entry point barrier could be sensual barrier. If you see that it is the food is putrid, the food is stale, or the water is dirty, it is your senses which are warning you not to consume it. Because of some of the bacterial decompositions, the food or the drink or the water may have some distinct smell, and by getting that smell you can immediately identify that this food or this water is not really good for consumption and you avoid it. So this could be the sensual barrier, and this is primarily for the defense of the body. The second barrier is the macro barrier, which is being governed by the skin, by the saliva, by the gastric juices. So somehow, if your body stick with the pathogens over the skin has a barrier. It doesn't allow the bacteria to get inside, or somehow if the bacteria are getting inside the body, your saliva, your gastric juices are having mechanisms to kill these bacteria and in turn to protect the body, or to protect the body from, or to defend the body from the pathogenesis. Now, if in case the pathogens are entered in your body, they have entered in the body, what the body is going to do with them? Naturally, because they are the pathogens, the body has to identify them and then has to treat them adequately. So the most important question at this point of time is how to recognize the pathogens. How to recognize these pathogens. This is quite interesting that the pathogens are primarily recognized by pathogen-associated molecular pattern. That is called as PAMP. For example, in this picture you see that this culprit is being shown with a particular pattern of dressing. So this could be a pattern, this could be a specific pattern by which a culprit can be identified. So similarly, the pathogenic bacteria are also having certain specific molecular patterns, which is being called as pathogen-associated molecular pattern. And by means of this pattern, they are being recognized that they are the pathogens. And now, who is going to recognize them? Okay, we found that they are the pathogens, but who is going to recognize them that they are the pathogens? It is recognized by the pattern recognition receptors, that is called as PRR, which are available on macrophages and natural killer cells of our body. So macrophages and natural killer cells are going to recognize the pathogens by means of pathogen-associated molecular patterns which are available on the pathogens, and these patterns are to be recognized by pattern recognition receptors which are available on macrophages and natural killer cells. Now let us see how do we recognize the pathogens through PAMPs. This is one of the most primitive methods of pathogen recognition. This identifies the pathogens which are most primitive in the history of evolution. That means primarily it identifies the prokaryotes, bacteria and viruses. And such molecular patterns are shared by all prokaryotes, and these molecular patterns have been preserved through billions of years. But at the same time, this do not recognize as the more evolved cells like eukaryotic cells or the cell cells. This is the depiction of PAMP in a bacterial cell. Many specific molecular patterns are available on a bacterial cell, may it be the gram-positive bacteria or may it be the gram-negative bacteria. It could be the cell structure, it could be the cell wall structure, it could be the lipoprotein or liponucleic acid, or it could be the bacterial DNA or lipopolysaccharides or peptide glycan or the protein which is available in the flagella of a bacteria. These all help the body cells, the macrophages and natural killer cells to identify the pathogens. And because these patterns are commonly shared by most of the prokaryotic cells, it provides the body a blanket defense against all these pathogens which share these common molecular patterns. So these are the things which are available in a bacterial cell and which help the body cells, body defense cells, to identify these bacterial cells. Now, when these molecular patterns are recognized by the body, there is a kind of molecular pattern and pattern recognition receptor interaction. There is a sort of interaction between the pattern recognition receptor, which is available on the surface of the macrophages or the natural killer cells or the polymorphonuclear lymphocytes. And this interaction leads to certain specific kind of events. For example, you see, these are the molecular patterns which are available on the prokaryotic cells. It could be lipopeptide, it could be flagellin, it could be peptidoglycan, which is being recognized by the pattern recognition receptor over a macrophage. This identifies it, then it engulfs it and it comes inside, it gets digested. And in the meantime, what happens? The nucleic acid of the pathogenic bacteria is preserved and it gets incorporated with the DNA of the macrophages. So in turn, it helps in production and secretion of certain chemicals, certain cytokines, which may be helping in the killing of the further infection of the bacteria, or they might also be having some bacteriostatic actions. So what this interaction actually does, we know that PRR, the receptors available on the surface of macrophages, dendritic cells, natural killer cells, polymorphs and mast cells, and they kill the pathogen by phagocytosis, and they initiate to secrete bactericidal and bacteriostatic chemicals, which may slow the process to get time for more sophisticated defense to get involved. This immunity or this defense which we had talked till now is the example of the innate immunity. So innate immunity, we can again summarize it that macrophages, the activated functions are the phagocytosis and activation of the bactericidal mechanism, or it can present the antigens. Dendritic cells can uptake the antigen in peripheral, can take up the antigens to the peripheral sites. Antigen presentation in lymph nodes, and neutrophils can phagocytose and activation of the bactericidal mechanisms. So these are the innate immunity responses. Now the question arises that what is going to happen if the bandit is more strong, new, more armed, sustained, and disguised? We need to employ a new set of defense subsequently to deal with this bandit, which is rather more strong, new, armed, and sustained, and very often disguised. So this new set of defense would be called as adaptive immunity. We would be talking about adaptive immunity in our next class, next lecture. Thank you. Thank you.